Good afternoon, everyone. Welcome to our first in the series on COVID-19 vaccines, specially targeted education program, which is aimed at building COVID-19 vaccine confidence by educating the educators. Just to remind you that this session will be recorded. And our first in the series is titled General Issues, Myths, and Misconceptions about COVID-19 vaccines. First, let's begin with a land acknowledgement. We would like to begin by acknowledging the land on which circuits operates. For thousands of years, it has been the tradition, the traditional land of the Huron-Wendat and the Petun First Nations, the Seneca, and most recently, the Mississaugas of Credit River. Today, Toronto is home to Indigenous peoples from across Turtle Island, and CIRCUS is committed to working toward new relationships that include First Nations, Inuit, and Métis people, and is grateful for the opportunity to share this land in caring for children and their families. So this afternoon, we are privileged to have speaking to us on this very important topic, Dr. Upton Allen. He's professor of pediatrics in the University of Toronto. He's the head of the Division of Infectious Diseases at the Hospital for Sick Children, CIRCUS. He's also a senior associate scientist in the Child Health Evaluative Sciences program in the CIRCUS Research Institute. Dr. Allen was past director of the Infectious Diseases Society of America, of which is a fellow. He's also a fellow of the American Academy of Pediatrics, the Royal College of Physicians and Surgeons of Canada, and the Royal College of Physicians in the UK. In 2018, Dr. Allen was awarded the Order of Ontario, which is the highest honor awarded by the province of Ontario. Dr. Allen, we welcome you and take the platform. Thank you.
Isaac, thank you so much for those very kind words, that kind introduction. And what I'm going to do, first of all, is to figure out how to share my screen. And let's see if that works. And you'll give me the thumbs up if it appears that my slides are working. Yes, okay. Okay, great. Are you seeing the first slide, Isaac? That's wonderful. Well, you know, once again, thank you so much. It's such a pleasure to be able to share some thoughts with you all today. The focus of my presentation is outlined here, where I will start by making some introductory remarks relating to history and the history of vaccines. And then I'll get into some specific questions that relate to what you see on the screen, who is behind vaccine development, the COVID vaccines, how were the vaccines developed so quickly. I'll also tackle some common myths and misconceptions relevant to COVID and more generally. And then some pediatric specific issues. As we embark on this series of our STEP initiative, pediatrics will feature prominently as we anticipate approval of the pediatric vaccines. And so some of the issues or questions that we see families raise are shown here. For example, interacting with families, they'll say things like, well, you know, can you tell me where are things regarding the autism issue of the past? Is this something that we need to think about? What about as additives or preservatives or things in vaccines? Tell us about that. The issue of multiple needles or multiple antigens. And what about long-term side effects, or side effects that don't come on now, but come on later on? And then what about if my child has an underlying chronic disease, be whatever that disease is, asthma, chronic lung disease, sickle cell disease, heart disease, can they be safely vaccinated? So we'll try to make this presentation focus a lot on some of these pediatric issues. But let's reflect on things somewhat historically. And we all, many of us in the field of medicine, nursing, and other disciplines often see this iconic image of Edward Jenner inoculating this young boy, being the first person to be vaccinated against smallpox in the modern era. This is actually Jenner's handwriting account of what he did. And I've just highlighted the time for you, May 1796. You know, there are lots of really interesting things in this. You can make out the words little chilly there. But you know, so he had a bit of a fever. So 1796. And then, interestingly, this gentleman, Benjamin Jesty, actually used cowpox for smallpox vaccination. He actually vaccinated his wife and sons, his pregnant wife, actually, in 1774. So actually prior to Jenner. So one might argue that he perhaps was the person who opened the door to the modern era of vaccination. But of course, Jenner has been credited with that. But then let's step back further, and there's a practice of variolation. In fact, in Asia, dried smallpox scabs were blown into the nose of individuals who would then get a mild form of smallpox. And then by about the start of the 18th century, variolation was used in Africa, India, and the Ottoman Turkish Empire. By 1716, this technique of variolation was brought to America by the African slave Onesimus, who introduced this to the Reverend Cotton Mather. And we're told that there was correspondence between Cotton Mather and folks in England prior to the introduction of variolation to England by Lady Wortley Montagu following her trips to Turkey. So one might argue that perhaps the introduction of variolation to England might well have been indirectly related to the work of an African slave. That's not a popular thing to talk about, but probably that's what's happened. And you can see this would have been a good 80 years or so prior to Edward Jenner. And this is a picture of Onesimus in New England. So a really important historical milestone. But of interest was that, and this is really important to think about, that as we run into issues relating to vaccine hesitancy today and lack of confidence, it's important as we interact with various individuals to note that this is not new. This relates, what I have on this slide relates to smallpox. But you can imagine if I were to read this to you, and wherever the word smallpox appears, if you say COVID-19, you realize, not much has changed. For some parents, the smallpox vaccination itself induced fear and protest. Some objectives, including local clergy, believe that the vaccine was unchristian. For other vaccinators, their discontent was the smallpox vaccine reflected their general mistrust in medicine. Suspicious of the vaccine's efficacy, some allege that smallpox resulted from decaying material in the atmosphere. Many people objected to vaccination because they believed it violated their personal liberty, a tension that worsened with mandatory vaccine policies. This was smallpox. You can imagine if you take out the word smallpox, it could easily apply to COVID today. And at that time, people promoted alternatives to smallpox vaccination, peppermints. It's amazing. Nothing has changed. But at the end of the day, it was recognized that a global effort was needed to eradicate smallpox. So you have children lining up in England, and the same thing in the developing world. This amazing effort led to the eradication of smallpox in 1978. Now, interestingly, one might argue, if during the smallpox eradication effort, we had access to social media, one wonders if indeed we would have eradicated the smallpox from the face of the earth. So let's get into some of the specific questions that often come up. One that we often get is, who is behind COVID vaccine development? Is this big pharma? Who are these guys? Is this all about making money? But there's some real faces of people that we know as scientists who are behind the vaccine development. Immunologist Kizzmekia Corbett worked at NIH with Anthony Fauci. She's been regarded by Dr. Fauci as being the person who developed the Moderna vaccine. She's now moved on to a role at Harvard University. And then we have this Turkish-German couple, Professor Shahin and his wife, Dr. Tureki, who founded a company, BioNTech. And they were working on mRNA vaccines for cancer, partnered with Pfizer. And this was leading to the development of mRNA vaccines for influenza. They pivoted and were able to apply the technology to COVID-19. A little known fact is the role played by a Canadian company, Acuitas Therapeutics, who developed the lipid nanoparticles that allowed for those particles to be used as a vehicle for the mRNA that we see in the Pfizer vaccine. So Canada does have some role in this development of the Pfizer-BioNTech vaccine. Judith Absalon is a Haitian-American woman. She has relatives in Canada, Montreal, and she led the early phase 1-2 trials of the Pfizer vaccine in the United States. Andrew Pollard, Canadian trained, University of Oxford scholar, just been knighted by the Queen for his tremendous work in developing the AstraZeneca vaccine. And as I understood it at the time, it was developed with a partnership that required that AstraZeneca wasn't going to make any profit from the vaccine. There are other people, Professor Mahdi from South Africa, who has led the AstraZeneca vaccines in the continent of Africa. And then there are many other individuals who have been people we know, who are faces, real faces, that reflect scientific knowledge and not just big pharma. How are these vaccines developed so quickly? And this is really an important point. What I often say is to think about the lessons that we have learned from SARS-CoV, that I call SARS-1 in 2002 to 2004, and the Middle East Respiratory Syndrome virus. It's important to note that the virus that causes COVID-19, SARS-CoV-2, is about 80% identical to the virus that caused SARS-1 in 2002, 2004. And it's about 50% identical to the Middle East Respiratory Syndrome coronavirus. And work on the SARS-1 vaccine started in 2003, 2004. And this is a publication in 2009. And it could easily have been a publication from 2020 or that focused on SARS-CoV-2, but this was SARS-1. And here it's talking about a spike protein. And the important point is that there's a lot of work that was being done. We have over 100 clinical trials on mRNA vaccines that have been directed at various forms of cancer. So the work on SARS-1 vaccines, the mRNA vaccine work on cancers, all of this led to information being available when COVID came along. So in summary then, the vaccine work on SARS-CoV-2 and MERS provided very useful guidance that led to the development of COVID-19 vaccines. In other words, the COVID vaccines, in terms of their development, didn't go from ground zero up. They were going from somewhere higher than ground zero, simply because of the previous work that had been done on SARS and MERS. Another way to put this is that if COVID-19 had happened in 2002, 2004, it would have taken us several years to develop a vaccine, and we would be in deep trouble. So it's a good thing that COVID-19 happened now and not then. And it's a matter of luck, to be honest, why we were able to have such accelerated vaccine development. Another important point in the vaccine development is that the phases were overlapped and not sequentially. Typically, one does the first phase. At each phase, one waits 6 months, analyzes the data, and then move on. But here, it was possible to overlap the phase and the phases. And one of the reasons why the phases were able to be overlapped is that there were lack of major safety signals. Because typically what happens is that if you experience safety signals, that creates roadblocks, and you have to pause, analyze what's going on, and then move forward. And this essentially was smooth sailing all the way through. So those are the reasons why there's accelerated vaccine development, of course, coupled with a lot of financial investment and the pooling of efforts and knowledge to enable the rapid development of these vaccines. But key take-home point is that the work didn't go from ground zero. It was facilitated by work that was being done before. Now, in terms of some of the common misconceptions or myths that we often encounter, I thought I'd just touch on the issue of autism, because the parents ask those questions, and I just want to get it out of the way, that the initial work relating to autism and the MMR vaccine was found to be fraudulent and retracted. So that really is the bottom line. And medical and legal experts all now fully agree that there's no evidence that vaccines are associated with autism. One of the things that we struggled with in the early years when people were talking about the association between vaccines and autism was that it was easy to say that there was no credible research showing any link. But parents often said, well, okay, you're telling us that there's no evidence, but can you tell us what caused autism? And now, in the last several years, we have seen great data and some fantastic data, some of which have been generated here by Steve Scherer and his colleagues, that show solid genetic evidence for markers, genetic markers associated with the development of autism. So what you can say to families now when they do ask you is that, no, it's not a matter of we don't know what causes it. We now have evidence of genetic factors associated with the development of autism, but vaccines are not one of those triggers. It's also often important to note that families will bring up specific substances, like thimerosal is mentioned, and it's comforting when you can say to families, yeah, I know what you're saying, but can I tell you that in Canada, the MMR vaccine has never contained thimerosal. So one can debate on, what thimerosal is and what it does, but it is actually used in multi-dose vials to prevent germs and bacterial contamination. But in the case of the MMR vaccine, it never contained thimerosal. So in summary then, the MMR vaccine used in Canada never contained thimerosal. And in fact, more broadly speaking, for pediatric vaccines, no vaccine made in Canada since 2001 for routine use in kids has ever contained thimerosal. The exception has been the flu vaccine, which we'll mention in a minute, diphtheria, polio, Hib. These vaccines, in fact, go back to 1997, 98, when thimerosal was removed from those vaccines. The multi-dose flu vials do contain thimerosal to safeguard against contamination of the vials. So in summary, what about the COVID-19 vaccines? Because families will ask that. I've gotten those questions, I've gotten emails. We can state clearly, thimerosal is not in the COVID-19 vaccines. What about additives that are present in some of the regular vaccines? For example, aluminum salts can be used as adjuvants. And there are over many years of work that would suggest that they are indeed quite safe. And indeed, when present in childhood vaccines, the amount that might be there would be about the same in food and breast milk and infant formulas. It's really important to put things in perspective. But what we can say, they're not in the COVID-19 vaccines. Formaldehyde inactivates viruses and toxins, about 10 times more formaldehyde in the infant's body at any one time, from and food and drinks and so forth, than there would be in a vaccine that they get. But for the COVID-19 vaccines, again, it's not in the pediatric vaccines. So important to get that point out. Are there special additives or preservatives in the currently available COVID-19 vaccines that I haven't mentioned? And so when the vaccines came on stream, one of the first things that we all did as physicians and scientists was to quickly look to see what exactly is in the vial. And the way I think of it is like a chocolate-coated raisin, where the mRNA is the raisin inside of the chocolate. The outer coating of the chocolate is made up of lipid, fat, sugar, salt, and water. And that basically is what the nanoparticle is all about. The nanoparticle that envelops the mRNA is like a chocolate-coated raisin. It's lipid. These are lipids that are known, salt, sugar, and water. There's really nothing else. There are no adjuvants, additives, preservatives of any special kind, and no antibiotics. So if things change, we would be the first to let people know, but I think it's really important to very transparent in terms of what is not in the vaccine and also what is in the vaccine. I think using the example of chocolate coated raisin, I find is often very helpful for families to get their head around what this is all about. Now, can COVID-19 vaccines plus regular childhood vaccines overwhelm a child's immune system? Families ask this one. This is one of the more common questions that we get asked. What I can say is that generally speaking, the immune systems of children and adults are challenged by millions of antigens each day that stimulate the immune system. For example, at a 2-month visit, there are about 34 antigens in all the recommended vaccines for that age. But here's the bottom line. This is summarized nicely by Paul Offit from the Vaccine Education Center at the Children's Hospital of Philadelphia. In theory, the infant's immune system have the capacity to accommodate about 10,000 vaccines at any one time. So think about that. In theory, you could give a child, an infant, 10,000 vaccines one time. Nobody would ever do that, of course. It's worse than a pincushion. But it's just to illustrate the fact that this idea that a kid's immune system is going to be overwhelmed by adding another vaccine like the COVID vaccine on top of everything that they've gotten is not based in science. And because of the tremendous capacity of the immune system, 10,000 different antigens or more at any one time. Are the COVID vaccines safe for people with autoimmune disorders? This, again, not just a pediatric issue, but also an issue in adults. We get asked this a lot. Can somebody with autoimmune disorders get the COVID-19 vaccines? And the answer is yes. And so there are some societies do agree that disorders such as arthritis, multiple sclerosis, other disorders do not represent a contraindication to the vaccines, and that there are no abnormal signals that have been detected following the millions of doses of vaccines that have been used. And these individuals are at high risk of having bad outcomes from COVID and should be vaccinated. What about people with weakened immune systems, the so-called immunocompromised individuals? Is it safe for them to be vaccinated? And more broadly, why might some vaccines be unsafe in people who are immunocompromised? So to answer the question of first, whether or not it's safe for immunocompromised people to be vaccinated, one has to think, well, in the first place, why would a vaccine cause a problem in somebody with a weakened immune system? This typically occurs in a situation where one is given a vaccine that is a live or live attenuated vaccine. So when you give a live or live attenuated vaccine, essentially what you're doing is that you're giving a mild form of infection. So that essentially what's happening when you give the MMR vaccine, you give the chickenpox vaccines. So those can be a problem in people with weakened immune systems, particularly those individuals who have defects on what we refer to as the cellular part of their immune system, the T cell part of their immune system. And so those individuals will have a problem handling live vaccines. To that end, what about the non-live vaccines? The non-live vaccines do not represent a safety issue. It's a question of whether or not they will work. So for the immunocompromised person, the concern is this vaccine, this live vaccine, going to work? Or is it that there's an issue of safety? There's no safety issue, but will it work? And that's the main concern with the immunocompromised individuals as far as a vaccine such as the COVID-19 vaccines. And here I'll share with you some data, and I want to leave enough time for questions and discussion. But I want to show you the next few slides that were shared with me by Dr. Jeff Kwong from ICES at Sunnybrook. And I was actually quite shocked when I saw it. So it looks for the coverage, the COVID-19 vaccine coverage as of October 3rd, according to chronic conditions for kids 12 to 17 years of age. And if you look at the various conditions, neuromuscular, cardiovascular, respiratory, hematology, including immunodeficiency, complex disorders, when you look at the underlying problem, and you look at the number of individuals with the problem, the number who got one or more dose, and you look at the fully vaccinated, you're looking at 63 up to 74%. If you look at the next slide, diabetics, asthmatics, who are at high risk, these are kids, high risk of having a bad outcome from COVID, people with other types of chronic lung disease, even cystic fibrosis, other immunocompromised individuals, people who are known to have bad outcomes if they get COVID. The fully vaccinated rates don't get greater than 80 to 81% for cystic fibrosis and inflammatory bowel disease. So we're looking then at people who are at high risk of having a bad outcome from COVID, having vaccination rates, adequate vaccination rates of 63 to 81%. And that's not good, because these are all conditions where the vaccines are not contraindicated. And so it'd be perfectly okay, and in fact, highly recommended that these individuals be vaccinated. So obviously, there's more work that needs to be done to try to get people aware of the importance of being vaccinated with underlying chronic conditions. But we'll see as we move forward, as the pediatric vaccines, if they're approved, we'll obviously need to do a lot of work to help to let people be reassured that the vaccines are quite safe to be used in individuals with these chronic disorders. Now, coming near to the end, what about late onset side effects from vaccines? This is a common question. What about late onset side effects? And you note I say late onset as opposed to long term, because that's another entity. By late onset, I mean you get vaccinated now and parents say, well, you know, I don't know what's going to happen 6 months from now or 5 years from now. And so this is an important point. What we can say is that in general, side effects that could cause long-term health problems are very unlikely following any vaccination, including COVID-19 vaccination. And what we know historically is that when one sees side effects from vaccines, they generally happen within days, weeks, not usually months, perhaps up within the first 2 months. So the side effects don't come on a year later, 5 years later. it doesn't happen that way. And I've been thinking about this for quite some time now to try to see if I could think of a vaccine that has been administered and then side effects pop up years later. And I still can't think of any. Maybe Isaac, I don't know if you could, we can talk about it. I just can't think of any that has happened that way. Many years ago, there were concerns about a very rare condition with the measles vaccine, but that was shown not to be the case. So bottom line is that the monitoring that has been put in place typically captures all the side effects that are important within a 6-week period. And in fact, in the case of the FDA, they'll add another couple of weeks just for a little bit more of an insurance. So important to let the families know that the side effects are ones that are not going to show up a year, 5, 10 years later, because we get that question a lot from families. Well, yeah, I don't know what's going to happen to my child in 5 years from now. So with that, I want to thank you very much. Look forward to the opportunity to answer any questions that you have. Thank you.
Thank you so very much, Dr. Allen. This was very illuminating. Now we're going to go into the questions and answers session. If you have any questions, please enter them in the questions and answers section, and we will address them as we come to them. So there's been a good question posted, and which actually is a concern to a section of the population. And that is whether fetal cell lines were used to produce the messenger RNA vaccines. Some people, for religious and other reasons, are very sensitive about this issue.
So Isaac, thank you so much. That's a very important question. The answer is, so the mRNA vaccines that are used in Canada now, so Pfizer and Moderna, the answer is no. The fetal cell lines were not used in their production. For AstraZeneca, the answer is no as well. For the Moderna vaccine, for the Johnson & Johnson vaccine, the understanding is that the adenovirus vector was grown up in a cell line. But I want to just come back to the issue of the mRNA vaccines and the role of cell lines. In general, cell lines are used, and in this case, these cell lines would have been generated originally under ethical conditions from the early 70s. They are used in experiments that are done prior to the vaccine development by researchers who might be studying different aspects of the work that could potentially lead to a vaccine development later on. And so if you are developing a drug, a medication, and you want to determine if the medication is toxic to cells, then you can use a cell line to see if the medication destroys that cell line. If you're wanting to see if the medication contains live viruses that were not previously identified, because viruses use living cells to replicate. Unlike bacteria that just needs nutrition, sugar, and water, viruses need a living cell. Then you can inoculate that cell line to see if it contains any harmful viruses. So that's where that type of vaccine production research might have been done by some researcher in a lab that probably leads people to wonder if that was used in the vaccine development of these mRNA vaccines. And the answer is no, that they were not used in the production of these mRNA vaccines. But speaking more generally, Isaac, I just want to put the whole cell line thing in perspective. I do agree that moving forward, one should look at other ways of doing the research without using any cell line. That and we have to be thinking about that. But I want to say I did have a look, Isaac, at some of the various products that have been developed using preclinical testing way upstream of the product being developed. And I don't want to, I'm afraid to list names, but what I can say is that someone did a project where they went to a pharmacy in the United States and looked at the over-counter medications in the United States in that particular pharmacy. And they could only find one where this type of work wasn't done. So in other words, the products that we use on a day-to-day basis have used cell lines at some point in the preclinical development. So I'm not defending vaccines, I'm just trying to put it in some sort of perspective.
Oh, that's very helpful. And Upton, recently we've gotten very substantial data that suggests that the immune response to the messenger RNA vaccines after 2 shots tends to wane after about 6 months. So a questioner is asking, from where you sit and with your experience with vaccines, how long is this vaccine effective for? And are we going to need boosters every 6 months to a year or repeatedly? What do you see possibly happening?
Yeah, that's a really great question, Isaac. We're still getting information on how long the immune response lasts. I think that when we think of waning immunity and we think of boosters, Isaac, we need to think of 2 situations, and so that we can communicate this to our patients. Number 1 is what I would refer to as completion of a primary series. So if you take a transplant patient or one of your heavily immunosuppressed cancer patients who gets 2 doses of the mRNA vaccine and they don't mount an adequate response, a 3rd dose for those individuals we would regard as series completion. So for them, their series is 3 doses. Your series and mine might be 2 doses. So that's a difference. And so like with hepatitis B vaccine, we have multiple doses. Chickenpox vaccine, we have 2 doses. So 3rd dose, therefore, series completion, that is separate from a 3rd dose because of waning immunity, where the 3rd dose then is given as a booster. It's quite likely that boosters will be needed in the future if there's still a lot of COVID around. But it is possible, we just don't know yet, if the boosters of the future might be such that only certain high-risk individuals would qualify for boosters, would need boosters. So for example, that immunocompromised person, maybe people with chronic lung disease, asthmatics, maybe above a certain age in long-term care facilities. And so you could see a scenario in the future where a year from now, I don't know that this will happen, but a year from now, the recommendation is, well, boosters are needed. We're only going to recommend for people 65 and above, people with immune deficiency. So it might be more selectively given.
Thanks, Upton. And still on that topic, there are some experts that wonder, because of the difficulty and challenges of measuring T-cell immunity, cellular immunity, that our dependence on antibody measures may not tell the full story about how protected we are from the severe forms of COVID infection. What are your thoughts on that?
Yeah, so far, when we look at the extent to which we are protected by any vaccine, the measures that are done, because they're just easier to do, are ones that focus on antibodies.
Yeah.
So if you take chickenpox for an example, Isaac, we do chickenpox antibodies, but the real protection from chickenpox is cell-mediated immunity.
Right.
And so the antibody is really just a marker that, well, if you have antibody, probably you have cell-mediated protection. So the antibody is a proxy marker for the more difficult test, which we don't do. So in the case of the COVID vaccines, there's that concern as well, where there could potentially be a disconnect between antibody and cell-mediated memory protection, and that somebody indeed might have a waning antibody, but their cell-mediated protection is still pretty decent. And so when that person, if that person gets COVID, even if their antibodies have waned to a significant degree, that they probably might not get severe disease because they still have some degree of T-cell memory.
Yes, that is a little reassuring. I think many Canadians will be pleased that the FDA at its latest meeting approved recognition of mixed vaccines. And so many Canadians who got a mixture of AstraZeneca and Moderna or Pfizer will be able to travel to the US because those two shots, though they were mixed, will be recognized by the FDA. But I'm saying so because I think they may have examined the evidence that actually those who got the mixed, the AstraZeneca or the vector vaccines, and I think Professor Pollard made a statement when he spoke at Grand Rounds at SECAS, that there is some evidence that the vector vaccines work more on T-cell memory immunity than the messenger RNA vaccines, and that a mixed vaccine sort of administration makes some good scientific sense. What are your thoughts?
Yeah, I do agree with Andy Pollard's comments that that there's no disadvantage in my mind, to be honest with you, of mixing a vector with an mRNA vaccine. We obviously need more data to suggest whether or not, if you compare 2 doses of an mRNA versus 1 dose of vector and 1 dose of mRNA, is there a day versus night difference between those? We need more data. There's some theoretical basis, some differences, as you've alluded to. But I think at the end of the day, Isaac, when you look at how the mRNA vaccines engage your immune system, and then you look at how the vector vaccines engage your immune systems, they're reasonably identical. They're pretty close. And I often say it's like the mRNA vaccine is like the postman delivering a message to your home. Postman delivers a message and then leaves. But the message is what you use to generate your spike protein and whatever you act on. The vector vaccines, you drive to the to Shoppers Drug Mart or wherever to pick up the message that the postman put there, and you take it into your home. And from then on, it's the same. So really and truly, the way that the mechanism of action of these vaccines work is such that the differences are probably much smaller than we think. And so I'm pretty comfortable. I've always been, as you know, comfortable with the idea that if you got a dose of a vector vaccine and then you got a dose of an mRNA vaccine, I think that's perfect.
Right. Very good. These are very clear explanations. You said earlier that SARS-1, for which Toronto was an epicenter for SARS-1. So Toronto had an experience with SARS-1. And you mentioned that it has almost 80% some similarity with SARS-2, which is what COVID is. What was the difference between the epidemiology of SARS-1 that made it peter out, whilst COVID is threatening to be a long-standing pandemic?
So, you know, that's a really great question. I'll back up and share with you a bit of an anecdote. Somewhere around about, I don't know, maybe about 2004, myself and my colleague, Jeremy Friedman, and someone else, we were invited to take part in media interviews, the training for media interviews, because, you know, we just were pretty green and people felt that it would be good for us to be trained. And they asked me what I thought would happen in the future as it relates to whether or not SARS-1 would come back. And my comment was that I don't see any reason why SARS-1 wouldn't come back, but maybe not necessarily the same virus. And maybe I should go back and read that tape again, because that's essentially what COVID is. It's really SARS-1 coming back, but with a different virus. That different virus has led to a difference in the epidemiology, where SARS-1 was an illness that largely was confined to institutions, to hospitals. A couple of exceptions, but for the most part, within a building. What it meant was that it was, although it was deadly, and you know, as you know, many of us, I was really scared that we were just not going to survive SARS-1, to be honest. I chatted among my colleagues at the time. I remember I thought we probably, you know, could die from SARS-1. It was within the walls of hospitals, and so you could contain it. Also, there wasn't a readily recognizable asymptomatic version that was being transmitted. Kids were milder, but still they got ill. With COVID, SARS-2, it was community epidemiology. The disease was spreading in the community. And most importantly, which became a game changer. And I remember when, you know, this happened, I sat in my office and I said, wow, you know, we have a problem. You know, my quote to my colleague was, Houston, we have a problem. Because, you know, you have asymptomatic spread of an easily transmissible virus. That's a problem. And that's what the difference is, where SARS-2, COVID, the virus, is easily transmitted through asymptomatic vectors. And that was not the case with SARS-1.
So to use the military parlance, it was more stealth.
Yes,
and quietly causing damage.
Yes, exactly. And it's just evolved to be a much more, you know, in virus worlds, a perfect virus in the sense that viruses, Isaac, as you know, they want to replicate. They need cells to replicate. They don't want to kill their hosts. They want to quietly interact with the host, replicate and transmit. So if you put them in human terms, that's the way they operate. But unfortunately, there are some hosts who are overwhelmed when they get infected, and they may have bad outcomes, as we see with many individuals who have had COVID.
We have one question in the chat, wondering whether, as an ID expert, you are concerned about community spread in stadiums opening up for sports while children remain unvaccinated.
Yeah, it's a great question. Yes, I am concerned about that potential. We obviously have to reopen at some point. We have to be prepared to pull back. Hopefully, we don't have. Yeah, but whenever you have asymptomatic people in a crowded environment, as in a big ball game, you can get transmission. The question is, will you get sustained transmission and multiple people being affected? And I think we just have to look at the numbers and reassess, but it's a really important point. It speaks to the need, though, as you've alluded to ensure that when the right time comes, children are adequately protected. And in fact, it's an opportunity for me to summarize a key point with respect, Isaac, to how we protect children. So we protect children two ways. We create a strategy to vaccinate them if they can be vaccinated. But in many situations, they can't be vaccinated because they have immune problems that cause the vaccines not to be effective, or the vaccines are not yet ready for them. And then we do a strategy number two, and that is we create a protective cocoon around them. We call that ring vaccination. And that protective cocoon starts at home. But it might extend beyond the home. And you could see it more broadly applied to the society as a whole.
Thank you, Upton. This has been very educative and revealing. I think we've come to the end. Just to summarize, we've been told that vaccines have been with us since the 18th century, and the reactions of people to the vaccines and mandates is not dissimilar to what we are seeing today, except that today we have the almighty social media, whereby both truth and misconceptions and lies can disseminate so fast and influence people. And that's why we are having these sessions. So I would like to thank all of you who have participated in this session. And just to let you know that another session will be following next week, will be delivered by Dr. David Burtz, and it will be on the types of COVID vaccines, how they work, how they are efficacious and effective. It will be a good follow-on to what we've heard from Upton today. Also, it is important that you give us your response and evaluation to these sessions, because that will help us introduce enhancements to the program as we proceed, because we do definitely want to educate those who educate, professionals who receive questions from the public, so that we'll be able to prepare our people to receive vaccines that are working so effectively to protect all of us. So please, the evaluation information, the link is in the chat box, and we would like you to complete them. You may receive emails as well asking you to complete them, and we really value your responses to those questions about the session. So on that note, I just want to thank all of you, and we look forward to seeing you next week. Please invite your friends, because I think these sessions are really of high value and at the highest quality. So we will educate ourselves so that we can be better educators for our community. Upton, thank you so much. And for the organizers behind the scene, thank you for your hard work. Thank you for participating. Bye-bye for the rest of the day.
You too. Have a good day. Bye-bye.